Okay, ma'am. Simran,
So
introduce her.
everyone, I again yes, ma'am. So everyone, I again introduce you to our professor Ritu Gopal. So she has retired after serving as faculty member at Department of Biosciences and Bioengineering at IIT Roorkee, India for about 40 years. During this period, she has also held positions as the head of department and emeritus professor besides undertaking several other administrative assignments. So after earning PhD degree from Physics Department at Indian Institute of Technology, Delhi, he has acquired 44 years of teaching and research experience in the field of molecular biophysics, in particular, structure, isolation, and interaction of ligands with respect to quadruplex DNA. He has extensively used NMR absorption fluorescence in circular dichroism spectroscopy for analysis of data, in addition to surface plasmon resonance, isotopic titration, and differential scanning calorimetry, molecular modeling, and cell-based assay techniques. He has guided 23 PhD students and over 100 of BTech MTech projects. She has published about 75 research papers in high impact referred journals, apart from delivering invited lectures and oral presentations in international and national conferences. She has undertaken about 10 research projects sponsored by Government of Indian agencies like DST-DBT, CSIR, DAE, ISMR, etc. Now I would request ma'am to kindly deliver her talk. Thank you, ma'am.
Yeah, I'm now going to tell you about DNA quadruplex ligand recognition, the second part using some sophisticated techniques. Earlier, in the first lecture, you have already been introduced to quadruplex DNA and its importance. Then in the morning lecture, I had told you about simple techniques like absorption spectroscopy, fluorescence, circular dichroism spectroscopy, and some other techniques, how to recognize a ligand which is generally a small molecule which binds to DNA quadruplex. Remember, the aim is basically to study the binding and to find out how it binds, how it can stabilize DNA quadruplex, so that its function is stopped. Because for many functions, DNA quadruplex requires single stranded instead of being instead of the quadruplex form. So if you stabilize the quadruplex and don't let it open and don't let single strand be open, for example, transcription will not take place. Some other oncogene promoters, the same thing will happen. Promoter will not be able to do its biological action. And this will bring in an action as desired. Now, so the main issue is that we must understand how the ligand binds to G quadruplex, how the complex is formed, what are the structural dynamic aspects, how can we study binding, and how can it then stabilize. So thermal stabilization of it is equally important. And equally important is we must do testing. We may be doing structural studies or spectroscopic studies, but we need to test them on actual cells. So either we take cancer cells or we take some other living cells whichever are appropriate and do in vivo or in vitro by using some techniques, use cell-based assays. I come back first to that, and at the end I will tell you about some of the cell assays and general aspects. Coming back to the ligand DNA G quadruplex complex, since I'm going to tell you about the structural techniques, there are two major classical techniques, X-ray and NMR. Here are some of the things, some of the studied complexes which are thoroughly studied and their structure is known. You can see RHPS4, porphyrin ring, TMPYP4, BRCA19 is again a trade name, nephrine diamide. These are some of the ligands, and the DNA which has been studied is CMYC, which is a promoter of oncogene, and HTEL stands for human telomeric. So the techniques that have been used is X-ray and NMR, and you can see there are a wide range of interaction. Some of them may be, some of the brown colored, which you can see on the slides, are the ligands. And this ligand may stack at the end, it may intercalate in between the carotids, it may do both, or it may even bind to the grooves, may interact with phosphates through electrostatic interaction. So there are many, many ways in which they can be found. I am now going to focus on nuclear magnetic resonance technique. Now this, I am particularly talking on proton NMR, or by which most of the studies have been done. And let's say this is proton NMR that I'm discussing, so it is H atom that I'm going to talk about. In a very, very simple way, and what does NMR mean? NMR means nuclear magnetic resonance. For example, if I put 11.7 Tesla of magnetic field, which is a huge magnetic field, then H will give resonance according to its electronic charge, mass ratio, and so on at 500 megahertz. We call such machines as 500 megahertz machine. So resonance will be achieved that. Basically, magnetic field will split the energy level of the nucleus into two levels, and photon will be absorbed and then it will relax back. And resonance will be there only and only, you will see this absorption when the intrinsic frequency of the proton in magnetic field by which it rotates around the magnetic field, it matches with the external one. So there is a resonance as we call it. So let's assume 11.7 Tesla, and then if this is the magnetic field, proton will resonate at 500 megahertz. So here is the proton or an H atom that we have in our chemical system. This is the magnetic field outside. But the point is, nucleus is never alone. Nucleus is always surrounded by electrons. So what does an electron do? Electron is negatively charged. Proton is positively charged. Even if it aligns, it will align along the direction of magnetic field, but the electron will, is going, being negatively charged, will align against the magnetic field. Then the proton will not see, this field may be very small, so the proton is not going to see the external magnetic field which the machine is providing. No, it will be actually reduced by this, because the field that this will see is the outside magnetic field minus the magnetic field produced by electron, which means instead of resonating at 500 megahertz, it will really resonate at something like maybe 499.98 megahertz. The difference is only this much. But if I want to do my tuning, the one way is I want to see it at 500 megahertz, I will have to supply extra field. So what we machines do is they will supply an extra field in a way so that it resonates at 500 megahertz. And why extra field? This is coming because there is an electron which is aligned in a direction opposite to the external magnetic field. Now, here is the crux of the story. The story is chemistry depends upon how electrons participate. All molecular interactions depend upon that. So, if electrons are important, then it's going to determine how the electron is there, where is the electron, what kind of electron it is it participating in bonding, it is participating in molecular interaction, and this is what information we will get. So basically it is the nucleus which is resonating, but it is the effect of electron that we will be seeing. A very, very simple way is, supposing you have something like TMS, trimethylsilane, or let's say you have methyl groups and a highly covalent compound. Highly covalent compound means the nucleus will be surrounded by electrons, it will be sharing the electrons, and they will be present. On the contrary, this means you are having, let's say, CH3 group, methyl group, okay, in your compound. So this is covalent. So that means you will have to supply lot of extra magnetic field so that you get resonance at 500 megahertz. Now, instead of this H being CH3, let us say it is NH or OH. What does nitrogen and oxygen do? They pull the electrons. So if you have something of this kind, which will pull the electron, somewhat electrovalent kind, instead of being covalent, and takes away the electrons, then it will resonance near 499.98 or rather 500 megahertz. So this means if I have H present as NH, it will give me resonance at one place. If I have H as methyl, it will be at another place. Now look at this chart that I have it. TMS is here somewhere, methyl and all are on this side. We call this as decreasing chemical shift. In our terminology, we call it upfield because we feel that a greater magnetic field has to be given. Now, coming to this chart, if you can see here, this any proton which is here in methyl or all, it will resonate at this frequency. If you are methylene, it will resonate here. If you are CH, less number of electron, it will resonate here. If you have aromatic, now once again, aromatic electrons are delocalized. So there will be a difference between aromatic CH and aliphatic CH. If aliphatic CH is here, then aromatic will be shifted towards the left of this table. Please see on this graph that has been shown, it's a spectra which is shown. On the rightmost, you will have methyl. You will have something like tetramethylsilane. Tetramethyl and methyl groups will be close to that. As you move towards left hand side, these are what we call as shift shielding. Electrons are shielding the proton from outside magnetic field. So there will be less shielding because there are less electrons. So CH2 will be somewhere here around, let's say, 1, and then CH will be at some position like this. The same CH, if it is aromatic, will be still here because electrons have been removed. If you are talking of NH and OH and so on. You can see benzene is here, benzene CH, whereas normal CH is somewhere here. And if you have something like NH and OH, it will be right on the leftmost side. So this is a scale that we have. We call this scale, this is basically shifting of frequency. The shifting of frequency has been given the name chemical shift simply for a reason that it is a chemical effect. It's an effect because of the surrounding electrons. So we call it chemical shift. So instead of plotting in all our spectroscopy, we plot intensity versus wavelength or intensity versus frequency. Here we make a plot. We don't have to say 499.99 or 500.01 like that. So we take the difference in frequency. So it is the shift in frequency that we plot, which means by the frequency which it has shifted away from the standard 500 megahertz frequency. So this shift will be, now coming back to this, so this is chemical shift, which is actually difference of frequency that we plot, intensity versus chemical shift. Now chemical shift is mapped on this side, on the rightmost part of the spectra. It is the one which is most maximum shielding and methyl kind of proton. Then methylene kind, then CH will come here, then aromatic CH, and then NH and OH. And our scale is opposite. We write this as 0. In fact, we always take a standard because we want to see in electrical these things, there can always be a shift for any reason, fluctuations in electricity and all. We always and always put a standard so that we can compare our results with each other. So the standard we use is tetramethylsilate. You can see TMS. This is four methyl groups attached to silicon. So tetramethyl silicon is another reason for selecting. It's an inert compound. It is not reactive. If you put another compound in your protein or DNA, we don't want it to react. So this is taken as 0 ppm because it has large amount of covalent electrons, maximum. So 0 ppm is that. Oh, my figure has shifted. Okay. So 0 ppm is TMS. Roughly, I will tell you, 1 ppm is where we get methyl, 2 ppm is where we get CH2, 4 ppm is typically the place where we get CH, and 6 ppm is where we get benzene kind of, which means aromatic CH. And NH and OH will come at 8 ppm, 9 ppm, 12 ppm. So they'll be farther off on the left side. What have we achieved? What we have achieved is any chemical reaction that takes place anywhere of any kind and how the molecule is having, whether it's having CH3 group, NH or OH, we will have for each of them a different signal because they will not resonate at the same frequency. Supposing I have a compound which is benzene, I will get one of this resonance. Supposing it has got on the side chain a methyl group attached, I will get one resonance here. If it has got somewhere in side chain NH group, then I will get one resonance here. So now we are working on atom level, NH, OH, and the moieties that we have. One more additional effect you have to see, when we have aromatic, it is shifted away because they are delocalized. So another effect is, if you have a proton with something above it, another H above it, it will experience upfield shift. Something on this side will experience downfield shift for a similar reason, because aromatics have ring current effects. So what we have got is, this is a standard scale available. You will find it easily. So there is a range. One more thing you have to note. that all methyl will not come at the same place. Methyl attached to some O group or something, methyl attached to another methyl and so on, there will be change. Which means, ideally speaking, this is TMS, which we take as 0 ppm. Around 1 ppm is the range. It could be from zero point 0.8 to 1.6 ppm. All methyls attached to different groups in different molecules will come in that range. So it's a small, small range. Once you get a resonance line, you take a spectra. For example, here is a spectra of some compound. So when we take this spectra, we'll see something here, and you can see some lines here and some here. Since we know what is the place, for example, if you are around 1 ppm, we know there is a methyl group which is attached. So in this way, there is OH group or NH group attached. So we get resonance for each one of them separately. Compare this with OD, optical density also. We get in OD and resonance peak because of tryptophan, tyrosine, like the full thing. Here we are getting according to CH3, CH2. So we are working almost on atomic scale. So one thing, the first thing which NMR gives you is from the position where we get resonance. This is actual NMR spectra of some compound. From the position and compare it with table, we can make out that there is a methyl group present, some 2, 3 methyl may be present, 2, 3 CH2 may be present. So according to the scale, we will be able to characterize. There are other things that we need for structure, and NMR will give us that. The next thing is this. This is, please see this, this is something like an alkane that I have drawn. CH3, and there is a single bond connected with it. And you know that in alkanes, 1H and HAB, this angle is theta, which we call a torsional angle or dihedral angle. This angle gives us different conformation. Supposing A and B, when we go along the line of the bond and they superimpose each other, we call it cis, CIS. We call it cis state in such cases. Okay, and if HA and HB are exactly 180-degree apart, supposing I move this and this HA is right over here at this point, then we call this as a trans conformation. Trans means HA and HB are 180-degree angle. So the angle that I'm talking of is this theta angle. This is the angle between HA and HB, these two bonds. If you see in alkanes, many conformers may be present. Since we have free rotations about bond, we may have cis, we may have gauche. Gauche is equal to 60 degree. We have gauche minus 60 degree, trans, and so many other conformations. Conformers, as we call it, alkane has many conformers. Conformers are not easily separable. Conformers, melting point and boiling point and all those properties, physical properties may be similar, quite close to each other. However, NMR will distinguish between different conformations. The point that it does is, supposing this H is there, this is only H, it's not coupled, so it will give you one line, one spectral line. This is an H, and this is another H, and attached through two bonds, exactly like this, like your alkane. I'm talking of HA and HB here. So the one blue and red, if you see, they are separated through three bonds, just as you have it in alkane. Now what happens is, this is having resonance of its own, this is having resonance of its own. I am looking at peak of this one, let us say the blue one. I am interested in the blue. What will happen is, it will see that H sometimes is aligned along magnetic field, sometimes it's aligned reverse to magnetic field. Why? Because H is undergoing its own transition. Sometimes it's in higher energy state, sometimes it's in lower. So part of the time it is adding magnetic field, part of the time it is subtracting magnetic field. Additional magnetic field it is giving. So as a result, what happens? This line, this H blue, which would have given you a spectral line over here, it will never feel that magnetic field. It feels a magnetic field which is slightly less and slightly more, both, because red color H is going through its transition. So as a result, this line splits into two lines. Half the line here, half of the time here, half of the time here. When you take spectra on NMR, steady state, and so on, you will see two lines. And this will be, if this H was not present, then this is its position. It will be around that as center, it will be split. This splitting is because of 1H and 2H. We call these as spins. in our terminology. So this is spin and this is spin, and we call this spin-spin splitting. The first splitting I told you, first splitting was not splitting, that was shifting, and it was chemical shift because of electrons, its own electrons. That was this hydrogen atom blue which you are seeing, its own electrons were shifting it. Now what I'm saying is, I'm not talking of hydrogen gas, hydrogen molecule, or even this kind of group. I'm talking of a big molecule, and maybe two bonds away there is another hydrogen in my structure. Then what? Then this H is going to add and subtract a magnetic field. So I will see two magnetic fields. Accordingly, this resonance instead of being here will be at two positions. Now these are two. Supposing you have methylene attached to this. Instead of being alkene, it is methylene which is attached to that. Two H are there. Now what happens is these two H, either both are up or both are down, or one is up, one are down, all have 50-50 probability. Okay. First position is both are up, they will add magnetic field. One position is both are negative, so both are down, spin down. So they will subtract the magnetic field. And two positions there will be in which the first is up, second is down, and the second will be in first is down and the this is up. In that case, there are two positions. So this means this line will split into 1 is to 2 is to 1. And what is J? We call this splitting as J. Similarly, I may have CH and I may have methyl group attached here. In that case, there are three. All three up, all three down, two up, one down, and one up, two down. So these are various possibilities. All of them have 50-50 probability. Eventually, I will have four lines. We call this as doublet, we call this as triplet, we call this as quartet, which means if H, this blue color H that you are seeing, if it was alone, it would have given one resonance line. Now, we are dealing with complicated molecules. This H may have another H nearby. It will show you a doublet. It will show as a triplet. This kind of thing will show as a steplet. So what? One thing bad, your single line has become complicated, but the good thing is it's giving us information. And what is this information that you are getting? The theory will tell you that this J, which is 3J, 3 is to, please see this one, J3, and in the curve also here on the plot, we are plotting J3. J3 means 3 bond away, this one. This is 3 bond away. So H and H, if they are 3 bond away, which is typically a conformer like what you have in alkane, then J3 coupling, we call it spin-spin coupling is called J, and 3 shows you that it is 2 H are 3 bonds away. And this coupling is a function of theta. This is what this is saying. And the exact equation is this one, which means if they are present in cis conformation, I showed you which cis means A and B are overlapping. If you see along the bond line, I will get this splitting to be about 12 hertz. They will be 12 hertz apart. If I have this angle like it is shown approximately 60 degrees, splitting is 0. If I have this angle as trans, this is about 13 hertz or so. So, and this is a function of theta. Now that makes the life very interesting. This means if I get a split, I will measure J value. I can put a cursor and measure what is J. I will put J here and I will calculate theta. I will come to know whether my alkane that I have is gauche or trans or cis or any other angle. I will be able to calculate angle. It's a tedious task when you have too many molecules, but that's what it is. Now go back to what you were told in the first slide when you, Professor Pamita was telling you that you have C3 prime endo, you have C2 prime endo, you have anti-glycosidic bond rotation. So those are dihedral angle and torsional angle. By this kind of technique, you will be able to calculate what is the angle. So you will get this actually confirmation of it. So you will get structural parameters. So one thing in NMR is you get a line, so you characterize it. But you know the question you will ask is such a sophisticated technique. I am dealing with methyl. I know there is a methyl present. There is an OH in my molecule. I am doing ethyl alcohol, let us say. So I have a methyl group. I have a CH2 group, methylene group. I have OH group. I will get 3 lines. Okay, and NMR will give me 3 lines. Now these lines will be split. and these lines will be split. So they will give me J values, and from J values, I will calculate what is this dihedral angle, conformational angle. So the first information I got is chemical shift, that is more for characterizing my H. It's a methyl or OH or NH. The second thing I'm getting is conformational angle over here using this J value. So J is what we use. Uh, Coming to the next, this is another thing that we use. What we do is, let me go back, sorry, to the first slide. Another thing that happens is, supposing this H is here and this H is here. These are two magnetic dipoles. So they will have dipole-dipole interaction. If you recall, you would have studied charge-charge interaction. If you have Q1 here and Q2 here, charge-charge interaction is Q1, Q2 by R square. Okay, so the interaction is Q1, Q2 by R square. In dipoles, the interaction is mu by R cube, mu1 by R cube, mu1, mu2, mu1 by R cube. So one dipole will have a field, electric field or magnetic field, mu by R cube. Let's call it mu1 by R1 cube. There is another dipole nearby. CH is a dipole, and there is another H. That will also have its magnetic field mu2 by R2 cube. R cube, let us say. So R is the distance between them. So the next interaction that happens, one, this H, see, you cannot help it. You have a molecule. This H, it's sitting in its magnetic field. And this is a dipole, and this is sitting in its magnetic field. They are sitting in their magnetic field. You cannot help it. They are few angstrom away. So few angstrom away means quite a bit of interaction. This interaction happens to be mu1 dot mu2 divided by r to the power of 6, because r cube and r cube get multiplied. So this interaction is what we call as NOE, nuclear Overhauser effect. Nuclear Overhauser effect, because of the name of the person who found it. Basically, it's an effect that if two H are at certain distance, then there will be an interaction between the two, which is proportional to mu1, mu2 by R6. Mu1 is fixed. We know what is the dipole strength of one H proton. We know the dipole strength of the other. The real thing is R. So mu1, mu2 divided by R6. This is the interaction, which means if interaction will be more if they are 1 angstrom apart, 3 angstrom apart. If they are 6 angstrom apart, interaction will be less. So this interaction will give us distance, so which means we will be able to measure distance between this end and this end. Now this property has nothing to do with bond. For example, let us say for this is a part of my G4 DNA. I bring a ligand here. Ligand is sitting here somewhere close by. It is having H. Now the ligand is close to this H in space. It is not bonded. This H belongs to ligand, one molecule. This H belongs to quadruplex DNA. These two will interact with each other through space. Through space, electromagnetic fields are there and they will interact, and this interaction is mu1, mu2 by R6. If I can determine what is this interaction, more or less, and quantify it, I will get distance between them. But mind you, I'll get distance between this and this also. This interaction, today we are able to measure with instruments up to 5 angstrom, you will be able to measure it. Now, it may be bonded, it need not be bonded. So, these are the three basic things that we do. Three basic things. What is characterization of our compound that we do? Second, that is chemical shift. Where is the position of my spectra? Number two is spin-spin coupling J. I will call it J to be brief. So we will get conformational angle. The third we call it NOE. NOE or nuclear Overhauser effect is R. It will give me distance between two H atoms. One more thing we do, since our molecules are very complicated, DNA and all, we have very specialized techniques by which we are able to do correlation. We call this as proton-proton correlation. We are able to do a lot of these correlations and lot of these distance calculation we are able to do. And we need some special double pulse techniques to be able to do automatically. Otherwise, we have to do one by selecting. There will be overlap of resonances and we have difficulty. So we have developed a lot of two-dimensional NMR techniques have been developed in the last so many years, by which automatically we will get this correlation. So I will just show you what the correlation looks like in a 2D NMR experiment. 2D NMR experiments had 2-3 pulses in it. So coming back to straight away applications, so what I'm going to use is, and what everybody, that's the, almost the minimum that one needs for structural analysis and one uses, the most common thing is characterization, chemical shift, number two J value, and number three NOE value. And we try to get the structure of the molecule with the help of that. So let's say this is a molecule I am talking, this is anthraquinone ring. Please note here there is a CH2 group here, CH2 group here, CH2, CH2, NH, OH, and so on. Another thing that happens is NH and OH, they exchange with water as the solvent very fast and they're not easily visible. So we use some special technique. So this is a mitogendron is the name of this molecule. This is an anti-cancer drug. It's basically an anthraquinone with two side chains. And these side chains are at this position. You can see this and this 5 and 8 positions. What does its NMR spectra look like? This actually we are taking this because this binds to quadruplex DNA and it gives reasonably good cell-based assays have found that it gives good, reasonably good activity. So as an example I'm taking. So for example, my molecule is this. So what I get is here is my molecule. And so what do I get? I told you methyls are here and methylenes are there. So I get 13 CH2 group, 12 CH2, 11 CH2, 14 CH2. There are 4 CH2 groups. The one which is 14CH2 must have been attached to somewhere to OH or NH, that's why it is shifted more on this side. These are aromatic protons attached to the aromatic 6H, 7H, and 2H, and so on. So these are these ones, this one attached to the aromatic part. And these are exchangeable NH, which I told you they exchange with water. These are called non-exchangeable in our terminology. So we have got NMR spectra of all of them. Now you can see this is split. This is actually a triplet. If you open it, you can see it here. It's a triplet. So it's a triplet because it is coupled to another H2. I showed you if it is coupled to 2, it will show as a triplet. So these couplings we can measure. If I measure these couplings, that means I will elaborate this, I mean expand this portion, put my cursor and measure J values. I will be able to get the conformational angle. So this is a very small molecule that I have taken just to show you what is. So this is chemical shift. My resonances of each CH2, each H actually. This H may be attached as methyl, this H may be CH2, this may be aromatic CH, or this may be NH or OH. Each one will give its own peak. First of all, it will be spread out. So from the region I know, oh, this is CH2, this is the region of CH2 and so on. Next thing is I can put a cursor here and determine conformational angle. Going to the next slide. This is a two-dimensional NMR spectra. Don't look at this, I mean, don't think it's just complicated. It's actually this one, chemical shift is this scale, and then there is another scale on the other side. So both are chemical shifts. See here, this is chemical shift, and that is also chemical shift. So actually we have taken in a way one spectra here and one spectra there in a way, and this is actually a 2D NMR experiment, two-dimensional experiment. The same coupling that you were seeing as doublet will show up like this. So since it is two-dimensional, so basically peak has been cut. So if you have a resonance peak and if it is cut, it will give you a circle. So this is the circle that we are getting. So actually these are two peaks which are doublet. You can see these two, green and blue. It's a doublet. So if you put the cursor at the center of blue and the center of green, you will get J. You will be able to get J. You can get J from here, from here, from here, and we calculate what is our J value. So I will get my confirmation. Another thing we need to do is, in these things, we need to know to which carbon it is attached, because I have CH2 attached to a particular carbon. So if this is all, both are proton here and proton here. We can have carbon here and proton here. So for example, this carbon is coupled to this proton. This carbon this belongs to this. This H2 belongs to this carbon. This H2 belongs to this. There are 4 CH2 groups and there are 4 carbons. So this carbon line, this is carbon spectra and this is proton spectra. We can place it in that way. So this CH2, its carbon is not this one. This is not its carbon. This is not its carbon. This is not its carbon. Its carbon is this which is in line with this. This is correlation. So we can do correlation. This is single correlation. If it is connected to its single bond. So this CH2 is giving peak here. This is the carbon of this CH2 only. It will not give a correlation with others because it's attached only to one H2. So that is what it shows. This, why do we do this? We do this because we have to recognize our peaks. We have complicated spectra. We also have HMBC, which is multiple bond correlation. This is single bond. See, CH2, which means C is attached to H through single bond. So this is single bond correlation. This is multiple bond. So those carbon which are 2 bond away, 3 bond away, they can also be seen here. And this is also something like 3 bond correlation, 1 bond. These are used for characterizing our carbon, characterizing our R, and automatically we have to do it because it will become a very lengthy process. This is nothing, as I told you, it's the same resonance peak because here on you will see them as this only in my spectra. This is a resonance line as you observe in OD or fluorescence. Imagine that it is cut from somewhere in the middle, then it will show you a contour. So this is that contour. All these contours that you see is actually a peak. Going to the next, this is a still more complicated. This is the NOE that I was talking about. This basically tells you the following. This says that, for example, this is 12CH2. It is connected to 11OH, 14OH, and it is at this correlation depends upon 1 over R6. So if I get small peak like this is a small peak, then distance may be 6 angstroms. If I get big peak like this, distance may be 4 angstroms. If I get still bigger peak like this is a small peak. So you can see intensity of these peaks is small or big, this is big, and there is overlapping also. See this peak circle. So intensities are different. These are very small peaks. You can see here and here, very small peak small here. So this basically we do integral of this peak. Integral is proportional to 1 over R6. Mu1, mu2 are fixed. They are the magnetic dipole moment of H and H. So no variation in that. Only thing is in our structure, distances vary. There are different distances. So what we do is we take integral of this and this integral is equal to constant divided by R6. So a big peak means these two protons must be close. See here, if you can see here in this fourth. Okay, on the right hand side, this here where I put this, it's a big peak, smaller, this is still smaller and this is still smaller. This means these proton, the proton here is away from this proton at larger distance or smaller distance. We have exact methods to calculate the distances. So these distances we will calculate. We will calculate conformational angle, and so we are doing the structure basically. So this is again the same, and you can see here very clearly. See, this is a small peak. This is a bigger peak. This is an intense peak. These are big peaks, and these are very small peaks here. Small peak means the proton which is in this line, this happens to be some guanine NH, and this is far off from this proton. So this is T7H1, for example, sugar proton, and this is at a small, high distance to this one. In this way, this is the NMR spectra of the complex. So this is my G quadruplex, and this is the ligand. So I have taken the spectra of ligand, which has been added to the G quadruplex DNA. Binding has taken place. So what do? We take DNA, add ligand to it. We want to know whether binding has taken place or not. We will use absorption, fluorescence, circular dichrysal spectra, and so on. And we will come to know from all these techniques or SPR, binding has taken place, bound complex has been formed. We will get K equilibrium and so on. Now we have to get structure. How to get structure? To get structure, supposing this is my ligand, I have to know where is this H, at what distance is this H from my sugar, from my base gonine, from gonine 3, from gonine 4, from gonine 2. So from each atom I have to calculate distance. Now this is manually very laborious, but there are ways of doing it. As I told you, algorithms and all have been developed. And so we will know all these distances from here. These distances, we put them in a table form. For example, here. This is short contact of MTX with this. We don't get contact if it is 8 angstrom away, 9 angstrom away. We don't get any correlation. So this is short contact, which means, for example, I have 11. This is the proton of my mitochondrion. Red is mitochondrion, black is DNA. So this is DNA. This DNA T1 H2 double from the sugar was at a certain distance from 11AH. It was at certain distance from 14OH, from 67OH of mitochondrion. So what are the distances of this NH, this NH, this OH, this H from G quadruplex? So from this G quadruplex, what are the distances? All those distances are here, which have been taken from NMR. And these are chemical shifts. Now, if you plot chemical shifts, one thing you can see straight away that this is serially done. All protons have been put here. The one which this one shift is more by binding. This is a shift due to the binding, the change. The shift is less here, which means the binding is in binding. These two are participating more. They must be near the G quadruplex DNA. They have shifted more. They will not shift equally because it is an asymmetric kind of compound. Imagine this compound has been brought near G quadruplex DNA. All distances of all protons here and all protons here are each is different, unique distance, and accordingly they will come at different, they will give you different chemical shifts. So these are different chemical shift plots, and these are short contacts which we can calculate distance. Once we calculate distance, then in the computer we draw the structure of the G cordon and feed those distances. Once we feed the distance in the graphics, it will draw the structure. So this is my mitochondrion. One is binding on this side of quadruplex and one was binding on this. And I got 2:1 complex. Okay, there were two sites, and it was binding as monomer in this particular case. And you know what? In absorption and fluorescence, I was getting a stoichiometry of 2:1. So things will match because it's the same compound you have studied by OD or fluorescence or NMR. Only thing is NMR is complicated relatively, but it will give much more information. It will give you the structure. Coming to the next, this is another. I think at one stage I told you dimer is binding, so actually dimer was binding. This is dimer. Two mitochondrion are aligned antiparallel to each other, and at one step both are binding. There are two of them here and two of them here, which means you are having 4:1 complex. And in this particular case, when we were working with it in stoichiometry, we got 4:1. In this particular sequence, it depends on sequence. I told you G quadruplex DNA. Here, four molecules of ligand were binding to this. And the previous one which I showed you is human telomerase. This is from protozoa, tetrahymena, and four are binding to this. So both are NMR structures which we got. Now one is binding, like we do by absorption, fluorescence, and so on. A very important aspect I told you, it should stabilize the DNA. More it stabilized, better it is. Okay, so you have to what actually people do is they synthesize many compounds, do a lot of testings and all experimentally or theoretically, and then ultimately they have to do experiment to find out whether it is stabilizing or not. Now, how do we do thermal melting? This is simply like melting of DNA. If you know duplet DNA, if you heat, it will become single strand. Exactly in same way, you take quadruplex DNA. If you heat it, strands will separate. So melting will take place. So you have to determine at what temperature was it melting when it was alone DNA and when it is complex. So these are the two things that you have determined. That means quadruplex DNA alone, what is the TM or T half as we call it, is the melting transition, melting temperature. And when the ligand is attached, what is the TM? So there are many methods and I will show you. One is immunoproton and one is absorbance, one is CD, but we use, and I think the best is I explain you with the straight away. These are four methods, and there is one more, differential scanning calorimetry. How we can use to determine thermal stabilization, which is a very important aspect of these interactions. Okay. So this is, I'm showing you now here, a simple girl. See here, this is alone and this is with mitosentron. So when it is alone, peaks were disappearing at 318K only. And when we put with mitosentron, peaks are visible at 318. They are still visible and still visible here, which means at least by these three temperatures you have stabilized it. This is an OD curve, only thing is we have plotted dA by dt. A is absorbance, and so it has given us peak. And this is CD. CD in quadruplex is different from CD in single strand, so melting has taken place. This is CD value along y-axis and x-axis is temperature. So we have done heating. Another thing we do is we do this temperature, we determine, you can see red and green and black. Black is a lone quadruplex, red is 1:1, and this is 2:1 complex. So 1:1 stabilized to some extent, 2:1 did more stabilization, and when we added 3, 4, much more of drug stabilization was the same. Reason? Only 2 were binding. So it has got thermally stabilized by this much of temperature, which is about 15 degrees centigrade here. Whereas in this case, 4 at D by N ratio of 4 only we got stabilization because 4 molecules were binding. I showed you the dimer. So simple OD, CD and immunoproton to show you. I'll quickly go over just few more examples. This is another we have done just to show you this is immunobinding and this is the structure you will get. And this is again the same OD stabilization and how chemical shifts change. So in different complexes we can do phosphorus NMR. Phosphorus NMR clearly one line will shift a lot. This is well known from theory. If intercalation takes place, and that is why we do phosphorus NMR. We did not get shifts in this case, which means for these ligands of ours, we were not getting intercalation, they were binding from outside only. So that is another, this is another complexation that is done. And this is another technique of thermal stabilization which has come up few years back. This simply depends the heat which is supplied to sample and resist. And excess heat capacity is measured. Basically, whenever thermal transition takes place, heat is given out or given in, and that is what you are measuring. So heat change is what you are measuring. Earlier, you know what we were measuring? We were measuring OD change, absorbance changes, CD changes, and now we are saying heat changes there. So this is another method of finding TM. So this is an instrument that one can use. This is the phenomenon. See, this is by immuno we were seeing, now we see here. And there are three transitions. You see, you can see two transition and this. It's not one transition. One transition should be one curve like this. So there are two, one, and you can see a hump here. So basically, when you convolute, there are two transitions taking place, which means there is an intermediate. Quadruplex is four-stranded. Four strands are there. When you heat, there are intermediate, partially folded, partially unfolded. When you heat further, there is maybe another partially folded state, and will still you heat, then you will get all the four single strands. Okay, so there are partial states in between. That is why you got three curves or this. So what do? We do alone quadruplex. We will do alone this or this quadruplex. And second time what we do is do the same experiment of the ligand which is bound to that. And that's how we determine how much it is. This is another one in which we have determined how much is the change at dn equal to 2, dna. All the complexes we have made, and you will see the lines are shifted there. There was a Tm of about 15 degree in this case. So what next? We don't have definite rules. We find that lot of stoichiometry differences are there. It depends on sequence, and the thermal stabilization is there. It is depends on that. So there are reasons is because different type of associations take place. This is another method that is used. It's very useful, though it is with Autodoc tools you can use it. So we have used it to determine for human telomeric and for both are human telomeric with potassium and sodium, and we have taken one such compound and taken on compound and see by docking which groups are participating. So by docking, we will come to know which are coming in contact. Now this is important. The experiment is always important, NMR or structure or whatever we do it. We cannot go on doing experiment. It will be a very, very laborious class and very, very expensive. So we need, what people do is they think of that, okay, this should be substance group, this should be the group, and they have 50 compounds in their mind that these are the ones we should try as for anti-cancer action. They cannot synthesize 50 and they cannot do experiment with 50. So first you must use such techniques to do docking and find out which gives how much of energy and good amount of contact. And then you try to, bring that 100 compounds which you have in your mind to 10 or 5, and then you can do experiment with that. So these are the results of docking. And this is another thing we do for binding isothermal titration calorimeter, which tells you how much is enthalpy change, entropy change of Gibbs free energy and equilibrium constant. This is also heat changes, heat bursts that you are getting. You get delta S in this technique. Now I'll show you why is this important, this delta S. Here. So we did delta S and this is one of this. So you come to know this is the way it is going. So this is the way it is going as you add more and more drug to your DNA. It's the same kind of compound. See the result is here. This shows that in particular interaction with this compound, delta S changed by minus 6 kilocalorie, delta S was negative, which means enthalpy was favorable, entropy was not favorable. Now you know that hydrophobic interaction are entropy driven. If stacking takes place, it is enthalpy driven. Each good stacking reduces the energy by 13 kilocalories. So your energy should go down by minus 13 kilocalories. It should show the change. Partial stacking may be minus 6, minus 8, like we got it. Supposing hydrogen bond is formed, again there will be, like in the first lecture she told you, what is the molecular association at atomic level? It's hydrogen bond formation, it will give you enthalpy change. If it is stacking, that is also attraction, it will give you enthalpy change. If it is hydrophobic interaction, it will give you entropy change. So entropy will be favorable. In some cases, lot of methyl such groups will come closer and hydrophobic ones will dominate. You can see in this case, entropy was favorable here. Entropy change was 12.90 kilocalorie. Ultimately, delta G is this, that is Gibbs free energy, and one can calculate K equilibrium with this. So what ITC is telling you is hydrophobic interaction may be dominant, hydrogen bonding may be dominant, stacking may be there. We may do all kind of these experiments by spectroscopy, calorimetry, equilibrium constant, and so on. We always and always have to test where, supposing I am making for telomere, then I have to test whether it is having anti-cancer action or not. Or I am doing some proto-oncogene, whether my cancer action has stopped or not. So cell viability assay or cell toxicity assay has to be done. This is a very standard, you will find MTT technique. In this actually yellow color MTT compound is where chemical compound. This becomes purple colored when we add some reagents to it, and it will become from yellow to purple if you have living cell, because living cell has some enzyme, it has got mitochondria, it uses that. So this is to find out whether my cells are dead or living. Literally it is that. So we take this and we add different concentrations of our drug sample. Supposing this is this is controlled. So then we add the drug into it and different concentrations and make this plot so that we get IC50 value, inhibitory concentration, where 50% inhibition has taken place. So we know the cells are getting killed. If the cells are not getting killed, then there is no point in going ahead. So this is some of the compounds which we have done, and the cells we have taken is breast cancer cell here. So we have taken breast cancer cells, we have incubated, and I'm showing you the results of that. So basically in the breast cancer and all this that we were showing, whether cells are getting killed or not. Cells may be getting killed, but the point is, I was working with telomere. Telomere had G quadruplex. Was that the reason? Maybe there was some other reason. So the first thing is I must see whether anti-cancer action is not there. Cell has stopped reproducing. So cells are not dividing anymore, which means no cancer action now. Okay, that is one thing, but I must make sure that it was because of telomeres. Telomere is the one which had that sequence which I told you, human telomere. So what we do in such an experiment is, this is telomerase repeat amplification protocol called TRAP assay. So we have extracted this enzyme telomerase from breast cancer cell line in this particular case which I am showing you, and then we add this, and then the ligand which I was doing, anthraquinone, 2,6-anthraquinone, and whatever you have in your mind, you put that at different concentrations. So one is a ladder, one is without control, and so on. and then you add different concentration, and then you can see here it stops telomerase, which means it is not replicating any further now. So it is because of G quadruplex DNA, because of the human telomerase. So this assay needs to be done to be sure that your compound is actually binding to G quadruplex DNA in vivo, in the living system, and that's how it is killing the cells. So you can see that this is the way it is going. IC50 normally we refer to the where 50% inhibition has taken place for the growth. So this is one you can see with another compound we did. This is another, and that's it. And one more slide I have to show you. Yes. So this is telomerase. When you are working with human telomere, you need to do this. If you are working with G quadruplex, which belongs to oncogene, then you will have to see that it is binding to the oncogene promoter, and that oncogene is not there. So you will have to do cell assay to ascertain. Another thing I told you at some stage, some bacteria and all. So another assay which I am going to show you, which was fortunately done in our lab, this Mycobacterium tuberculosis. This is the bacteria which causes TB disease. So TB disease, Mycobacterium tuberculosis. So in that disease, what happens is we found that in this particular one, there are few genes which were having GGG, repeated genes. And it was forming quadruplexes. So there were some studies which showed that these genes were having G quadruplex, they were having repeated G. So what we did is we took some same compound mitocentron and we took one amodin. Amodin is found in aloe vera plant. So we took mitocentron, MTX and amodin and we tried to find out, this is inhibition assay, minimum inhibition. So we have done with MTX, with MO and minimum and this. Again it's the same, pink color becomes blue color and so on. So we find out whether this is killing bacteria or not. So our compound, whether it is killing or not, so inhibition assay is what we have done, like your MTT assay. So this is only thing is bacteria we have taken, tuberculosis bacteria. Okay, this has been done in collaboration with Indian Institute of Science, Bangalore, because tuberculosis bacteria we cannot grow because you need some kind of protection and all that, and they have developed labs for that. Similarly, like in India, everybody cannot grow HIV virus because then the infection would spread. So this is in collaboration with that, and so they work on this bacterial mycobacterium tuberculosis. So we found that some genes were having it. So we found out by assay, first of all, whether it was killing bacteria or not. In this case, what happens is there are two genes which do transcription. They are very important for the respiration of the bacteria. The two genes we found here, this and this, they had negative. This is RT-PCR we have done, reverse transcriptase polymerase chain reaction. Basically, it is a reverse transcriptase we have done with and without our drug. So these are the things with and without drug, and we got very good negative activity. See, it has become negative. So it stops the transcription to a larger. If it is negative beyond -2, it is considered quite good. If you ask the bacteria people, they will tell you that two-fold change is quite good. We got three-fold change and more than four-fold change in some cases. And we found that it was stopping the transcription activity. We did CD spectra and so on. We did not do NMR. We have already published these two papers also. So what I'm trying to tell you is what all can be done. See, HIV virus is having some G quadruplex. This is having, few years back I saw a report malarial parasite is having, which means and if the function can be stopped. So you will have to do biological assay, as I said, you will have to kill the bacteria or you will have to kill the breast cancer cell lines. So in the cells must get killed, and then you will have to show that it is binding to G quadruplex. So ultimately, what next and what do we have? Some drugs have been developed, like for example, carfluxacin here, another drug, Braco 19, like this. They, some of them lot of pathogenic bacteria have it, lot of virus. I gave you an example of TB virus, tuberculosis. HIV also has it. So there are many bacteria, virus, which also have proto-oncogenes have got, besides the human telomeres. So there is a lot of scope. One needs to do bioassays and one as I said, two, three are already undergoing clinical trial. Braco 19 is one, quadfluxacin is another, which is based on G quadruplex DNA. So good luck to all of you, and I hope the research in this field keeps going on. And I also hope at the end that I have given a glimpse of various kind of techniques which you our structural studies, dynamic studies by spectrometer, calorimetry has to be coupled with cell-based assays. And then only it would make a sense and the drugs can be developed. Thank you very much for your patient listening. If you have questions, we can take. If you have something specific later on, you can take email ID from the
It was, I think, I would say the complete course in 2 lectures, compiled in 2 lectures. I would request, okay, ma'am, I have a doubt because
yeah.
one thing, because as you know, we have been working in this area from last 15 to 20 years. UV visible and fluorescence titration that only let us know that how the conformational changes they are a complete idea about the atom to atom interaction. However, I'm still confused because recently there is some finding. They say that if you reduce the fraction of sp3 hybridization on the molecules,
Yeah.
has to bind to the quadruplexes, the binding is better than the class of anthraquinone antibiotics.
Maybe it is possible. See, one thing more I'll tell you. Everybody cannot go into structural techniques, and structures
Yeah.
are not easy to solve also.
Yes.
They're very intricate. See, NMR itself, somebody who has been working for 10, 20 years, he can only solve NMR structures. So
Yes.
these are very specialized techniques. Actually affecting in cell-based assays, it may still be very good enough. Toxicity should not be there. It should not affect the other parts in your cellular system. So it is still possible. You need not know too much of detail of it. And there are people who are working on that, as I said. So it is possible that you have this. So there are some linear chain compounds, and as you said, sp3 hybridization or something is important. It is quite possible. The which has gone into clinical trials, the problem is they have some kind of other effect on your cellular system. That is another thing you have to see. You are doing isolated experiments in lab. Doing it on animal system or human system and then other things connected to it is something very different. Very different. So, but there is lot of scope in that because we can see, as the years pass, more and more information is coming. And it was only, imagine, it started only with telomeres, human telomeres. And then came proto-oncogene. And now it is virus and malaria. Malaria And another thing is we did not have techniques to study interaction also, no? That also you have to see. That is also a thing.
But
So
they are saying that we do have a non-telomeric G quadruplexes also.
yes,
We do have that.
tuberculosis, mycobacterium, and one is responsible for NADH synthesis and so on in the metabolic processes. So the gene is called NDHA and MOSR. So these are two genes which we targeted. There are other genes. Somebody has published, two more genes are there which somebody has published.
So can it be taken as a molecular marker?
As a molecular specific, wherever there is quadruplex DNA, it's binding just to that.
Yeah, but
Yes.
that's the thing.
Those are being developed.
Yes. So means if you want to make it a more target specific, so
Of binding to quadruplex versus duplex. You will come across such papers also. But there are papers, which show you that the same molecule like mitosentron, which was binding to duplex, is binding to
yes,
quadruplex. This is
quadruplex. So,
also there. But
yes. So,
the effect is different. Let me tell you, binding constants are different, effects are different, structures are different. So one
but
will
this
have
is
to see the cell-based assay.
Needle's work. He has
Yes.
specifically said after such a long time because he said that yes, if we increase the factor of sp3 hybridization going to be quadruplex specific only. So I think
Actually,
that would help in target specific designing of the molecules.
right. He actually has done by X-ray crystallography. He is an X-ray crystallography, and their research group synthesizes lot of molecules they have done. Lot of papers they have published on that. And they did only this, from there only, by the way, we have taken 2,6-diamido anthraquinone. We have taken from there. They did with pipridyl, morphylyle. Only thing is they did not do some of the studies which we have done by NMR. The corresponding. So
No,
they are able to pinpoint, like they took aromatic ring, pipridyl, this, and they also took diethylamine and monoethylamine. So they are able to pinpoint, like you said, whether CH3 group is a more potent molecule as compared to the pipridyl or morphinyl. It
see,
is
this is
actually the molecule which you have seen, which I showed you. We did NMR and all. They did not do NMR and so
no,
much of
they
stuff
don't do.
like
They
that.
simple do
But
synthesize and they make
basically,
all cellular studies.
it is their compound. In fact, somewhere we even somewhere my student forgot to acknowledge that, and he said we are the ones who did trap essay of that. So I said yes, you did.
No, actually, one of his co-worker, S. Bala Subramaniam, he specifically in one of his reviews has said this. He is also there, but I think his affiliation is from ISC and he is now in Oxford. He specifically said that in order to make it quadruplex target specific, we need to have फ्रैक्शन ऑफ sp3 कंपोनेंट मोर सो वो एंथ्राक्विनोन वाला फ्रेगमेंट मींस उसमें एक और जैसे कि हमारा माइटोसेंट्रॉन में डाइमिराइजेशन हो जा रहा था डाइमिराइजेशन
Very
मॉलिक्यूल
bad.
हाँ सो
It's not good.
डाइमिराइजेशन में इट विल नॉट बी एबल टू मे बी
Accommodate also.
या
It cannot be accommodated easily.
yeah so that type of thing because if the molecules start dimerizing itself in a before it interact with the receptor then may not be having maximum interaction maybe having more side effect maybe having more side effect So
So
this is something means without NMR, without X-ray, I don't think so. We would be able to,
they
judge.
have worked on monoethyl and diethyl also. Monoethyl and diethyl added in the side chains for this purpose only.
Mhm.
That's good.
Thank you so much, ma'am. I could. We are really blessed to have you. Thank you so much, Simran. Please over to you, Simran.
Thank
Yes,
you.
ma'am.
So much.
It's a lot of work actually.
Yes, ma'am. I know it's still like
Part of it. I think it's about the work I have mixed, I have presented. The results are from our work. Theory remains the same.
I know.
It's about 8, 10 research scholars.
No, it is only after 2016. Before that, so I know it is
Yeah.
maybe three times more.
After that, again, 4 more PhD students. And this Mycobacterium tuberculosis is my last student.
But this work I have not means I didn't know that this last slide which you discussed with us, so I didn't know about that.
Uh, I can send you anyway the publication, the list, and you will be able to find all.
Thank you
It
so
was
much.
very interesting, but the point is, even I was getting disappointed. It's happening at such a last stage where मेरी हिम्मत नहीं है आगे काम करने की. And the student is now in Austin. She's in University of Texas, who was very enthusiastic, and she used to go. And another thing, sometimes it happens, by the way. That is my student. He did MSc from IIT Roorkee, and he was my student, and his wife is also my student, Amit Singh.
Uh-huh.
His wife is also my student. She did project. Means both are MSc from IIT Roorkee, but he did PhD with the Nobel laureate and postdoc in USA. He is doing very well. He got Bhatnagar Award also recently. He has been asked to set up this lab, and he is working on Mycobacterium tuberculosis. That's why we had access to all this. Otherwise, how can we think of growing a tuberculosis? It needs very special facilities and all, no? So he had those type 1, type 2, type 3 facilities. And readily he said, nothing doing, I mean, for you, whatever you want, you tell us, because they were growing that bacteria for some other purpose. So doing an inhibition for them, if I give them a drug, it does not matter. For RT-PCR also, it's hardly anything. They are looking at other genes. So I told them, these are the 4 genes you try. He tried. It was not a big thing because they were already culturing that bacteria.
Because this requires a
Mycobacterium.
very specialized lab. We just cannot have it, anywhere.
No, we need permission from government of India because you cannot grow virus
Yeah,
like
yeah.
that.
Yes, yes, yes.
So he has that. So I was fortunate. He just came to for something and came to my department, say hello, and then he told me I'm working on that, and I just told him. And it just happened. And then the only thing I was saying, you have come at such a last stage, even I never diversified my interest. So we could have done much more. Anyway, whatever we could do, we are happy.
Thank you
We
so much, ma'am.
contributed.
Thank you. It's really
Well,
blessed. We are really blessed. Simran, over to you.
best
Okay.
wishes to
Thank you,
all
ma'am.
participants.
Thank you so much, ma'am, for such a nice talk. I wish you all found this lecture very useful and informative. So I want to take a moment to sincerely thank each and every one of you for your active participation and enthusiasm throughout the session, throughout the day. A special thanks to our esteemed instructors, Professor Ritu Barthwal and Dr. Aliza, who shared their expertise. So with that, I formally bring the today's course to a close. Thank you for once again and best wishes to everyone. We will meet again tomorrow at 10 a.m. for our next speaker talk by Dr. Pamita Avasthi. Thank you.
Goodbye.
No, it's not my talk tomorrow. My talk is day after.
Sorry, by Dr. Jagannath. Sorry.
Yes, 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 yes. So
Okay, sorry.
tomorrow at 10
Tomorrow
p.m.
talk will be
Yeah.
from Dr. Jagannath.